Hey everyone, Connor Wander here with the Straight From a Scientist podcast, and I am super excited to bring you this new series. That's right, series all about Alzheimer's disease. So to give you some context, I was in San Diego for the last week of October and the first week of November. It was an excellent trip, and I was there for both the Alzheimer's Disease Fast Track Conference put on by BrightFocus, which is a research and advocacy nonprofit focused on eliminating, curing Alzheimer's disease and glaucoma, two major degenerative diseases that we just really need to fix. So Alzheimer's Disease Fast Track, I'll call it AD Fast Track, is a satellite conference to the much more well-known Society for Neuroscience conference. AD Fast Track had about 100 guests. You can see the picture of a lot of the guests in the show notes and the speakers as well. And whereas Society for Neuroscience is multi-thousand, 28,000 people attended this year alone. So very different conferences. We're actually going to have guests in this series met at both of them, but the majority of the guests will be coming from the AD Fast Track Conference. So that's going to be the focus for this series. We've done only a couple episodes on Alzheimer's disease throughout all of our podcast history, even though that is my research focus. I tend not to talk about it too much, but what you can find is in episode 14, Maya Gastilla, who is an undergraduate, a postbac researcher at the NIH right now. She's applying to grad schools, I think, probably as we speak. So good luck to Maya. We talked about how amyloid beta may actually have a protective role in Alzheimer's disease. That's episode 13 if you want to check it out. However, we're going to have a lot more discussion of that and amyloid beta's possible role in the amyloid cascade hypothesis and how exactly we think amyloid beta might fit in to the ultra complex cascade that is Alzheimer's disease. So the general mood for the conference was humble. There was definitely a lot of humility at the conference because we realized as Alzheimer's disease researchers that we have made many mistakes in the past, and we have been perhaps a little overzealous in our claims that a cure for Alzheimer's disease is only a few years away. And recent failures in clinical trials have had people feeling a little hopeless, perhaps. And in the research field, have people feeling like we need to backtrack a little bit and do some soul searching, if you will. There's definitely a lot of valid criticism of the Alzheimer's disease research field. But the mood was not just humility at this conference. There was a lot of hope. And that hope, I think, stems from recent advances in technology that allow not only new methods of direct analysis, but also technology is bringing researchers together in ways that it never could before. We're going to hear about these new technologies that are setting the stage for major breakthroughs in the field, some of which have already happened, some of which are right around the corner. And then we'll talk a little bit about the implications for the field at large. Also, this series is a great opportunity for you to really get to know some of the top names in the Alzheimer's disease research field. I cannot believe that BrightFocus was able to bring so many influential researchers to the forefront. And not only that, but a lot of them agreed to talk to me. So I'm super excited again to bring you this series. Hope you enjoy it. It's going to be very different from our typical interview format, but we're going to have 3 episodes and they'll each have at least 3 guests, which I will break down for you. One last thing before we get started, I really want to thank Rachel and Martha from the BrightFocus Foundation for helping me organize all of these podcast interviews, whether they were during a coffee break or after dinner or at random times all throughout the conference event. I couldn't have done it without them, and they helped me organize the show notes as well. So huge shout out to them, and of course the BrightFocus Foundation, everyone involved in putting on that conference. This episode guests in order are Dr. Diane Bovenkamp, who is the VP of Scientific Affairs for BrightFocus, and she's going to talk a little bit about what BrightFocus does, how they help the Alzheimer's disease research field, and just a little bit about their overall mission, as well as the AD Fast Track Conference. After Dr. Diane Bovenkamp, we're going to hear from Dr. Frank LaFerla, who I think puts Alzheimer's disease in a really great perspective. Not only does he help us understand the link between amyloid beta and tau, which are the 2 proteins that have been long identified to be involved in Alzheimer's disease, but then he also talks a little bit about how Alzheimer's disease might just be part of a much bigger problem with our society, with the way we live as modern humans. And we talk a little bit about some of the ways that he goes about structuring his lifestyle. And finally, we'll hear from Dr. Bruce T. Lamb from Indiana University, who along with Dr. LaFerla and many other universities and companies, is heading up the Model AD program. So that's an acronym, and he'll explain that in his segment. But Model AD is essentially trying to build new animal models for Alzheimer's disease. This is part of the reason that there's a lot of humility in the conference, I think, because there's been a lot of talk in the news about how animal models don't necessarily replicate Alzheimer's disease. But there are a lot of new advances in technology, like I said. In general, a bunch of great ideas from a bunch of great scientists are all coming together into one massive effort that I think is going to bring the field closer and closer together. And that's what we need. So with that, I will begin the segment. Enjoy. Welcome, everyone. I'm here in the conference room of the BrightFocus 80 Fast Track meeting, and I have with me the VP of Scientific Affairs, Dr. Diane Bovenkamp. Dr. Bovenkamp, thank you, first of all, for setting this whole meeting up, being part of that, and second off, for talking to us.
Well, thank you for sitting down. Well, first of all, for coming as a participant, and then having an interest in interviewing everybody. So
happy to do it. It's been, like I've said in other segments, it's been really helpful just for my own personal gain. But BrightFocus is much bigger than just training new students and new postdocs. What's the overall goal of BrightFocus, if you could sum that up?
Yeah, so our overarching goal is to try and eradicate the diseases of mind and sight. So in particular, we're funding innovative researchers to for Alzheimer's disease, glaucoma, and macular degeneration. So we really think that that you guys are the future, the people who are attending Fast Track right now. You're the future, and we want to try and help to guide you to come into the Alzheimer's field and then thrive. And ultimately, the people who we talk with every day are the affected families, and if there's some kind of benefit we can give to them, that's ultimately your goal as well as researchers.
I love the BrightFocus tagline, by the way: cure in mind, cure in sight.
Absolutely.
And so it's the mind being the Alzheimer's disease component, and then sight being the glaucoma component, which I actually don't know too much about the disease, but it is an amyloid-related disease similar to Alzheimer's, right?
Well, it happens that there are protein deposits in macular degeneration. You have the drusins that deposit in the retina and in glaucoma. You know, one of the proteins that are labeled as a bad protein in glaucoma do clump together. But yeah, no. So the eye is an extension of the brain, really. So if you can't really have a eye transplant because the retina is part of it's a nerve. So there are a lot of common features of that perhaps people can learn lessons from one disease and bring them together. We actually have I also organize another one-day symposium that happens before associated with the Alzheimer's disease, Parkinson's disease meeting. That happens every 2 years, and we're actually going to have one that's going to happen in Portugal in March, and it's all about bringing scientists from all three of these disease fields and trying to see if there's common ground. For example, ApoE. ApoE4 is a risk factor for Alzheimer's, and ApoE2 is a quote positive factor for protective for Alzheimer's, but it's the reverse for macular degeneration. So why is that? And so some people are starting to look at that too.
Yeah, it's really interesting disease crossover. Like Dr. Manikov mentioned the other day, how if you eat some amount of this toxic fruit, you could get ALS, but if you eat a little less of it, you might present Parkinson's-like symptoms. So watching that crossover, understanding of that crossover evolve is really interesting. I think throughout the conference, it's been super exciting just to, one, see and meet all these experts that I consider like science celebrities from my end.
They're rock stars, right?
Yeah, exactly. And to be able to do that as a student of my stage is really appealing. So I'm sure other people are going to hear about this and want to get involved. What are the eligibility criteria for being an attendee at the BrightFocus 80 Fast Track meeting?
Yeah, the BrightFocus Alzheimer's Fast Track. Actually, so we're actually going to go. Normally, we have been having it every 2 years, sometimes every other year. We're planning on. We kind of follow it's an official satellite meeting of Society for Neuroscience. Mm-hmm. So we're going to have it in Chicago next year, and really, it's first come, first serve, because, we want it to be an intimate experience. As you know, we split up into 7 groups for the mock grant proposal challenge that actually you're going to participate in later this afternoon.
Yep.
That'll be exciting. But yeah, so we don't want it to get, bigger than maybe like 60 or 75 people. So this year was, as the word spreads about how awesome this meeting is, this is the first year that we had to close off people being able to sign up on the website. So maybe in the future, what we might do is have people sign up to receive an e-alert. But we send it out to all past grantees and our scientific review committee, and try to get the word out. And as I've talked with many different attendees, I've always been asking them, well, how did you hear about us? And it's like a friend of a grantee, but it's great. Yeah. But thank you for getting the word out about this. So.
Yeah, of course. I mean, I only found out about it because my PI sent my the postdoc in my lab, who's also here, and myself a link. So without that, I mean, we wouldn't be here. As soon as I read the speaker schedule, I knew I had to come, but
boom.
Yeah, exactly. So I just signed up, and I wasn't even going to SFN before that. So
yeah,
as long as I'm in San Diego. But we've had a lot of great speakers. We've had a lot of great demonstration of technology and new techniques. Is there anything particularly that you're super excited about, or any trends in the field that you've seen emerge over the past few years? Like, what are you most excited about in the field of Alzheimer's disease research after the past couple of days?
Yeah, well, I mean, it's kind of hard to choose. I mean, when everybody, we did our, everybody got up, all of the attendees got up, and actually most of the speakers did like a 1-minute elevator pitch, which I think was really effective this year. You really have to condense what you're doing down into 1 minute. And I think, it's everybody is working on a piece of the puzzle, right? And so ultimately, you don't know exactly, because it's such a complex disease. Dr. Alzheimer, first described it in 1906. So this has been around for quite a long time. And there've, as you've learned from the speakers, there have been a lot of, disappointments. We thought we had the aha moment, and we're going to have the cure in 3 to 5 years, as Dr. Eisen was saying, and then you had another turn. So, but yeah, that said, I think that one of the foremost in my mind, what would be really exciting is to have a blood-based biomarker for early detection. And I know that really the 7 assignments that we gave you were actually what I consider to be really, really exciting topics. So we have the sex-based differences, we have the inflammation, we have vascular contributions. So I'm actually really looking forward to to what the students, what the attendees come up with. But yeah, no. So the way that BrightFocus works for a grant application for Alzheimer's disease, we actually have 3-year, for our standard awards, we have 3-year grants worth $300,000. So that's not too bad. We actually have postdoctoral fellowships we just raised at $200,000 for 2 years. And we kind of leave it in the old style of not putting out a directed call because we think that will help, maybe some of the ideas over the years that attendees have kind of hatched up here, or kind of had a jump off point from talking here. they came and applied to BrightFocus for funding, and our scientific review committee found it to be really exciting. So that's, part of what we want to do is catalyze the ideas, and hopefully maybe you'll be applying for a fellowship at some point in the future, but anybody who attends here.
So that is a huge component of the BrightFocus goal, right? Is to support research with your grant program. I didn't really get any of this, and I think we'll get it later today. We're going to have a big discussion on the grants. But just for a quick snippet, is there any sort of strategy with the grant selection program? Are you looking for some of the more innovative projects, maybe the high risk, high reward ones that could, not every other grant agency might fund, but is going to have huge potential for impact? Or are you looking for something that's just really personally exciting? I would love to know a bit about that process for everyone who's potentially applying.
Yeah, well, you said it all. There you go. Drop the mic.
Sorry, I don't want to.
No. So we do have a process. We have a scientific review committee. I'll just talk about, we have 3 committees, for the 3 programs, but I'll talk about the Alzheimer's disease research. Right now, it's led by co-chairs Dr. Holtzman, Dr. Zhang, and we get some of the best minds in the world to critique and score and look at what is the most innovative research. So for a lot of foundations, for us, it has to be, we welcome people coming in from other fields, but it has to be directly related to Alzheimer's disease and related dementias. And as you'll see in the process today, we're actually going to try and do the process very similar, where people apply. You don't have to write something up with this, but you have to do your 15-minute presentation. Then we will be asking you questions similar to questions that get raised in our in-person meeting, where there are two or three primary reviewers that bring forward and describe and say their scores, but then ultimately your grant application you have to convince an entire committee that this is something that is important to fund, because it gets discussed. There's people revisit scores, and then there's a range. And some people, say if you have a range from 2 to 5, if you use the NIH score, then maybe some people might go from one or the other based on that discussion. So when you're writing your grant application, you want to make sure that you convince an entire committee. But ultimately, we end up funding a lot of those innovative early stage ideas, almost like an angel funder or venture capital, right, organization, to be able to develop enough preliminary data that then, if you did receive this funding, then you could apply for, to an organization where you could then further develop that idea, like NIH or a pharma company. So, who
typically fund more safe bets, yeah.
Well, not necessarily, but they sometimes they require a higher burden of proof. So if you just have a really cool, innovative idea that maybe you're hatching here, you don't have any preliminary data for it. So we'll help to catalyze and kickstart that.
Very neat. Where can people find the application portal? Just on brightfocus.org?
Yes, brightfocus.org, and if you click on there's a grant section, and it'll have every little bit of information there if the portal's open. So right now we're closed for the Alzheimer's disease applications. That just deadline was October 2nd. If you want to apply for glaucoma, it's still open, November 9th. But yeah, so if you go on to our website, there on the left column, it'll say sign up for e-alerts. So if you put it in your email address there, we will let you know when the request for proposal email gets sent out. For Alzheimer's, it's about in July that we send it out for submission the next October.
And we'll also, of course, toss a link for everything in the show notes. Thank you so much for coming on, Dr. Bovenkamp. Is there anything else you'd love to say to our listeners?
Yeah, well, I mean, I'm so excited that I get to be that's another thing, that we don't fund In the U.S., it's global. So we've actually funded 20 people in 22 countries. And actually, funny enough, we actually have people who have originated from 22 countries who are represented in this meeting today. I mean, not necessarily the same countries. So to find a cure for Alzheimer's, there's no geopolitical border. It's international. Any idea is, I think, will be important. And in terms of the attendees at Fast Track, I'm really excited that at the end of today, I'll be able to welcome everybody as an alumni. And I mean, a part of the reason why we have it connected with Society for Neuroscience is that now I think all of you will be well prepared to go and wander the halls and confidently talk with some of the leaders in the field and some of your teammates, your alumni here, and who knows, I've heard that many alumni do meet on an annual basis at Society for Neuroscience moving on, so maybe it'll be interesting to see what this group does too.
Yeah, I expect it fully with this group as well.
Yeah.
Thanks
All right.
again, Dr. Bovenkamp.
Thanks so much.
You're great.
Bye.
Welcome, everyone. It's a bright and sunny morning in San Diego. We are actually out on the balcony this time, so we might have a little bit of ambient noise, but it's a great spot to talk. And I have with me Dr. Frank LaFerla. Dr. LaFerla, thank you so much for coming.
Thank you for having me.
So Dr. LaFerla is one of the organizers of the 80 BrightFocus Fast Track conference here, and he's been with it for a long time. So we're going to talk about that in a bit. Probably one of his biggest claims to fame is the triple transgenic model of Alzheimer's disease, and it's a really good model for helping the field advance its understanding of how amyloid plaques and tau tangles, the two protein deposits that we see in Alzheimer's disease patients, work together to induce the progression of the disease. So people have very different opinions about how those two proteins work together, and these mouse models are a great tool for helping us get there. Now, I actually use the triple in some of my studies in the Song Lab, so it's very near and dear to my heart. What was your experience with that model, and how do you think it's helped you understand the potential link between amyloid beta plaques and tau tangles?
Okay, so let me give you a little bit of background first, if I may.
Yeah.
So when I was finishing up graduate school, I decided to move into the Alzheimer's disease field, and I thought it was important to try to develop animal models of human diseases. And at that time, the technology was so expensive to do that, and it was largely restricted to a handful of laboratories in the country. And so I went to one of these labs learning how to develop animal models. And as with many technologies, with time, it gets cheaper and cheaper, and eventually a lot more people are able to participate in the field. And so I had a two-pronged strategy for my career. And the first was that I realized that developing animal models probably would not help me get tenure, and that I would focus on development of animal models for the second part of my career, which was trying to have a major impact. And so with that in mind, the field was, I think, a little surprised that the amyloid or APP expressing mice didn't develop the full complement of Alzheimer's pathology, and that was very different than what we anticipated early on. And we thought that since the genetic evidence was so clear that mutations in APP would lead to the full-blown disease, that the same thing would happen in a mouse. And I guess one of the biggest lessons we learned early on is that a mouse is not a little human. And so it became very clear that one needed to take much more aggressive strategies. And at that time, people in the field were doing this by doing multiple crosses, taking APP mice and crossing them to tau mice. And I started thinking if there was an alternative way to do that, because it was very time consuming and very expensive. And so what we decided to do was make a transgenic mouse from a transgenic mouse. And so we started off with these presenilin mutant knock-in animals, and we used that as our base. And early on, since I had made a lot of animal models, recognized that when you made these, very rarely did you get integration of a single copy of the transgene at a locus, and so thought that, well, maybe since the APP and tau transgenes were roughly the same size, maybe they might integrate at the same locus. And so that's exactly what happened. And that was very fortuitous, because it meant that even though our mice were triple transgenic, they essentially bred like a single transgenic line. And so it was very easy to propagate them. And so it gave the field a tool in which you could very quickly grow up a large number of mice and age them, which was not something you could do readily by crossing.
Yeah, the logistical limitations of so many experiments are really what are holding us back in a lot of areas, especially in the Alzheimer's disease field. Now, a lot of discussion lately about the technology really brought us to that point. But I thought you mentioned an interesting thing right at the beginning of that is that normal APP seem to not be able to turn tau pathological. Is that the same today with all of the other APP models that have come out since?
Yeah, I think it is. And I think, part of the problem that we have inherent in mice is that their lifespan is, 24, maybe 28 months. If you're lucky, you might be able to push it to 32 months or so. That might not be sufficient to, trigger that cascade of events. And so the mice might need a little bit of assistance, which you could provide by, either introducing other human genes. Yeah.
And so those other human genes, in the case of the triple, were tau. How do you think that progression, what is that missing link? Is there even a link? I'd love to know your thoughts on that.
Missing link in terms of
amyloid beta and tau.
Yeah. So a lot of our studies suggested that they seem to be very closely linked, and that one of the outcomes of our studies, at least in the triples, was that amyloid seemed to be driving the tau pathology. And so no matter what we did, either genetically or pharmacologically, if we raised A beta levels, we seemed to get more tau pathology. Conversely, if we lowered A beta, we got less tau pathology. But when we would focus on manipulating tau levels and either genetically or pharmacologically raising or lowering tau, it seemed to have no impact on the A beta levels. And so to us, that suggested that A beta lied upstream of tau in the pathological cascade, which I think is consistent with the human data as well.
And people often refer to that as the amyloid cascade hypothesis. It's something that I've invested a lot of reading into, and not everyone agrees with, which is an interesting thing to see at this conference. And of course, everyone is targeting a different element of that chain.
Yeah, and I think if I had to give you my 2 cents on that, I think a lot of people believe that amyloid is the trigger for Alzheimer's disease. I think the genetic evidence is so overwhelming. But what we don't talk enough about is that just because something is the trigger doesn't necessarily mean it's the best therapeutic target. And it certainly would be a great therapeutic target very early on in life. But once someone transitions to that disease process, then it no longer becomes as attractive a target. And I think someone yesterday at the BrightFocus meeting said that think of amyloid as the trigger on a gun, and tau is kind of the bullet. And so, if you use that metaphor, once the trigger, you're not going to be able to cure people or have much therapeutic value if you focus on the trigger once it's been shot, right?
That's fair. Yeah, it's a fair point. That was actually Dr. Henry Sang. He's a postdoc in my lab. So the paper he was referring to is by George Bloom. It says amyloid and tau, the trigger and bullet in Alzheimer's disease pathogenesis. And I'm constantly going back to that review and looking at the citations. So, but moving past kind of the basic understanding of the disease, the protein cascades that we've just been talking about, we had a really interesting quick conversation the other night, and you have a really cool take on just kind of the afflictions that are hitting humans today and how Alzheimer's disease might be part of just a bigger problem, if I can extrapolate on that.
Yeah, so clearly we're living longer, right? And so some of the problems that used to face mankind before are no longer as problematic as they once were. I think I was mentioning to you that I was reading this book called The Brief History of Mankind by Yuval Noah Harari. And, he makes some interesting points that, in the past, humans were impacted by famine. And now, for the most part, and it's not entirely true, but for the most part, you're more, if you go hungry, it's because your government wants you to go hungry. There's no excuse for anyone in the world to go hungry because food is so plentiful. And then second comment he had was that most people died from infectious diseases. But now with the advent of antibiotics and other treatments, that's not the case, and if anything, we're actually maybe too clean. Right. Right. And his point is that in today's society, you're probably more likely to die from eating too many cheeseburgers than from an infectious disease. And with all this stuff, there's it's mainly true, but there's clearly exceptions. And then the third point was that a lot of people used to die from wars, and he gave reviews a lot of the statistics, and I think he said that from like 2012, there were about 150,000 people worldwide that died as a result of war or any terrorism, and somewhere on the order of about 500,000 people died from crime, 600,000 people died from committing suicide. But what was shocking was that about 1.5 million people died from being diabetic, and so his conclusion was that sugar seems to be more lethal than violence in some ways. So I mean, obviously it's said with being tongue in cheek, but I think it just shows and highlights what's happening to our society. And so you look at our life extension, and it's just been pretty phenomenal, right? I mean, the fastest growing segment of our population are people over the age of 90. I think if you're born after the year 2000, which you may be pretty close to if you're not there.
92, not that close.
Not that close. But if you're born after 2000, you have a greater than 50-50 shot of living to your 100th birthday. Time magazine recently put a cover of this cute little baby on it, and the caption read that this baby may live to 142 years of age. Yeah. And so I think all of us, we don't only want to live longer, we want to make sure we live better. Right. And that's the key. And unfortunately, a lot of these age-related diseases, Alzheimer's, other dementias, really, impact that.
So there's the whole idea of health span versus lifespan, which we've kind of discussed at length in the conference. A lot of people have presented on exercise, yoga being very good potential therapies for Alzheimer's disease and just natural aging, which also there's that debate of whether Alzheimer's disease is pathological or normal aging. Now, given those suggestions that exercise and things like that are really beneficial in these states, and knowing as much as you know about Alzheimer's disease, do you focus on exercise at all? You mentioned yesterday, yeah.
So I'm a pretty avid tennis player. I try to play somewhere between 5 to 6 times a week, and I like it for obviously for the physical activity, the competition that you get out of it. But I also find that it markedly reduces my stress levels.
Nice.
And I just thinking about yesterday, sitting in this conference all day, just sitting in a hotel chair. But by the time I went back to my hotel room at 6 o'clock, I was wiped out.
Yep.
And I'm just thinking to myself, why do I feel so wiped out? I mean, I just sat there, right?
Yeah.
And a normal day at work, I'd be running around, running up and down the steps, running from one side of the campus to the other. And I just think that physical activity just gives you so much more energy, right?
And a stress resilience, yeah. It's my one complaint about this conference is that it's so concentrated and such a long conference. Like, I'm getting a ton out of it, that the gym hours are completely the same hours as when I have to be in the seminar area.
That's right.
So
exactly.
Yeah, that would be the one thing I would change. But other than that, I've loved the experience. What's been your experience with the BrightFocus 80 Fast Track Conference?
So I've been a part of this Fast Track for probably a decade now. I started off as a speaker here many, many years ago. Actually, I think we started off in San Diego when I was first invited. And somehow, I don't know how this happened, but I became a co-organizer of the conference. I'm just trying to figure out how that happened, but it's been a pleasure working with the staff at BrightFocus. They're incredibly dedicated, and my two co-organizers, Harry Steinbush and Cindy Lemire, are totally committed to this. So it makes it a lot of fun to put together, this program for the, junior investigators.
And everyone seems to be really friendly with each other, and people are making friends, and it seems like you all are old friends, if I've seen anything.
I think Bruce Lamb said it very well yesterday, and and I really liked the way he turned around and said to all of the students, turn to the person to your left, turn to the person to your right. Remember those faces, because you'll be in the field with these individuals a long time. And I think that's one of the remarkable aspects of this BrightFocus Fast Track is the networking opportunities that you can get, not only with your own peers, but with the faculty that are here as well, right?
Absolutely. So speaking to the future of Alzheimer's disease research, what are you excited about, and what are you watching, basically?
Yeah, so I mean, for my own sake, from my own point of view, I am focused on developing the next generation of animal models, and that's something that the NIH is funding as part of this Model AD consortium that we're in cahoots with Indiana University and Jack's Lab and Sage. And so that's one thing. I think the animal models will get better. I think they'll be more predictive. It's going to be a long haul. It's not something that's going to happen overnight, but it will be the right thing to do for the field. I think you heard from Josh Grill's talk that we're really spending a lot of time understanding the barriers to clinical trials. So I think we will have better clinical trials in the future and better therapies. I mean, we just have to keep going forward.
Yeah. Well, we're hoping for the best, obviously. Again, I really appreciate you coming on to talk, Dr. LaFerla. Is there anything else you'd want to say to the listeners?
Yeah. Just realize how critical scientific research is. It's really important for everyone to help support it. I often think of a quote that Carl Sagan had, that he said that we live in a society exquisitely dependent on science and technology, in which hardly anyone knows anything about science and technology. And in this country, we don't produce a lot of products anymore except our intellectual capital, and I think it's absolutely critical for readers to really support scientific research. That's going to be absolutely critical for our national security.
Yeah, and to add on top of that, I think anyone can fit the mold of a scientist. There is no one stereotypic mold. So I think if someone saw you in the street, they probably wouldn't expect that you're this super prestigious scientist. Is there anything else that you do that kind of you think gives you a good angle on your work that people wouldn't really expect a scientist to be doing?
Well, I give a lot of lay audience talks, and I really enjoy interacting with members of the community who really don't know much about science. And if I could just say one other thing, and that's to congratulate you for what you're doing with this podcast. It's really wonderful, and it's very much needed in our society. So thank you for doing this.
Yeah, I appreciate it. Keeps me sharp. So thanks, everyone. Welcome, everyone. It's Thursday morning, and I'm speaking to Dr. Bruce T. Lamb from Indiana University. He's going to be speaking later on different mouse models of Alzheimer's disease, but we're going to get into why we need different models of why we need different models of Alzheimer's disease, and what some of the challenges are when building those models. So a lot of focus lately, especially in the news, has been on the failure of clinical trials. Now, I think a lot of that owes to misdiagnosis of AD, and we need to make better biomarkers for the disease. But also, the mouse models that we're using in preclinical research aren't necessarily reflective of the whole population. From my own literature or reading, I can see that a lot of these mouse models are familial AD, which is only 1% of the population, give or take, right? So that seems to be a problem. But Dr. Lamb, if you could just give us your thoughts on.
Sure. Yeah, and I've as a way of introduction, I've been working in the area of animal models of Alzheimer's disease for my entire career. So about 30 years of working in this area. So I'm very aware of all the issues that have evolved with the animal models. And I do agree. I mean, I think the recent failures of a lot of the clinical trials, especially those focused on amyloid, have led to a lot of sobering conclusions and trying to figure out where does the field need to move. And I think there's several things. One, I think the failure of the clinical trials could be due, as you suggested, to a lot of different things. So, for example, the early anti-amyloid therapies, it turns out they only had about 20 or they had 20 to 30% of the population that didn't have amyloid in the brain to begin with. So obviously that's not a good strategy. But the new clinical trials are now enrolling to make sure they have amyloid before they move forward, and they're targeting earlier stages of disease, because that's one of the other ideas about why they were failing, was we need to potentially move earlier.
Right.
And so, but one of the other certainly ideas that was out there was, well, just as you said, all the preclinical data to that point had been done in animal models that had these rare familial autosomal dominant mutations that cause Alzheimer's disease in people. And really, they're all largely focused on amyloid production and deposition. And those I would argue, have been great models for what they are. And I would argue they're good models for studying amyloid pathology and how it progresses and all the biological pathways involved. And so that's been, I think, tremendously helpful, and has helped, lead to some of the current clinical trials, like the anti-amyloid therapies that are in clinical trials.
Right.
But again, I think we do have to take a step back based on some of the most recent data and suggest where do we go now. And so at the Alzheimer's Disease Research Summit in 2015 that the NIH had, one of the recommendations that came out of that summit was that we need to generate better models, especially focusing on late onset disease, where the disease is much more complex. So that's the other piece, that early onset disease is what we would call a fairly pure disease. So it's just, amyloid pathology followed by tau pathology. I mean, it's a very, what you see in the textbooks.
Right.
And then the late onset disease, though, is really complex, because most of those patients have multiple different comorbidities occurring at the same times. They have diabetes, they have you name it, hypertension, they have, and it's all vascular problems. There's a large diversity, I would argue, in that patient population. And then, so out of that meeting in Bethesda for the research summit, these recommendations came out, and then NIH put out a call for NIH proposals to sort of develop a consortia to basically develop new late onset disease models, to characterize them, and really with the goals of assessing as much as we can of how close we're getting to the human disease at every level. And then continuing to refine those models and hopefully get closer and closer to the disease, and even actually do some preclinical testing. So actually start to bring some therapies in, test them in the models. And then the final piece is to make all of that available to the community as fast as we possibly can. So we applied for that, and now there is a consortia. It's Indiana University, where I am, the Jackson Labs in Bar Harbor, Maine, University of California, Irvine, including Dr. LaFerla leads that group, who's here at this meeting, and then Sage Bionetworks, who are sort of a data analytics and storage group.
Very cool.
And so we've been working now about 2 years. We're about 2 years in. And really the idea here is we're over the entire length of the grant, we're supposed to generate 50 new models.
A lot.
Yeah.
For those who don't know how long it takes.
It's a lot of work. But the other big thing that really, I think, came online just a little bit around the same time was CRISPR. So the ability to do genome editing at both the speed and cost at which you can do that makes this now actually feasible, unlike before, where it would have taken you 20 years to make that number of models. And so we've been proceeding to make these new models. So the first sets of models, we made a new ApoE allelic series for mice,
which is the most or it's the highest genetic risk factor.
Oh, far and away,
right.
Yeah.
Yep.
I mean, it's the biggest late onset genetic risk. There have been previous models for ApoE, but again, they were difficult to access because usually there's so many restrictions put on them that people couldn't get them. So we decided we would just make independent ones and then just make them freely available to everybody. Yeah. And then we made another gene variant in TREM2, which is an immune receptor.
The second highest, right?
It's the second highest, yeah. And then now we have those that have double mutations, have both ApoE4 and TREM2, the R47H alleles. And the idea is we're going to continue to add genetic risk as it's identified, and then sort of move those forward. And so the entire consortia consists of a bioinformatics and data management group who was looking at the data out in the literature, but also coming from these huge studies like the Alzheimer's Disease Genetics Consortia, the Accelerating Medicines Partnership, AMP-AD data, MOVE-AD. So there's a lot of these big consortia that are generating lots of genetic data as well as omic data. And so we're trying to take the bioinformatics, we're trying to take all that information and come up with a prioritized list of genes and variants that we should be targeting in animal models. And then the second stage is to do a disease modeling project, which is a group of scientists who are really then targeted with first generating the models and then doing the phenotyping. Again, which we have generally two stages. One is we're doing a fairly fast and trying to sort of assess, are these models we want to move forward with or not? And a lot of that is using omics to compare back to the human disease. And then the second is a deep phenotyping, where we do everything from animal imaging to a large number of different omics, including transcriptomics, but proteomics and metabolomics, as well as biology, obviously, and biochemistry and biomarkers. So it's a pretty thorough investigation of these models. And we're doing that at multiple sites because the exact same models are going to be at the Jackson Labs, and they're going to be at Indiana University. Some will be at UC Irvine. And so we'll have data from each of those sites as well, which I think is a really unique advantage as well, because one of the things is in the past has been everybody has their own model in their lab, and they study it and get different results than somebody else at a different lab. But again, since we're doing this and then all the data is available, people will be able to assess how it works in their lab compared to what the data.
That's great. Open source science is going to bring us along.
Right. And then the final piece is a preclinical testing core, which is take the most promising models that have moved out of this disease modeling project and start to move therapies into those models. And that group has already generated, now trying to get nominations from the scientific community as what therapies should move forward. And we've developed sort of a web-based portal where we request information about the therapies that people are nominating, everything from what information is known about the PK and PD and all of the pharmacological data, just different information. And then that generates basically, again, a prioritized score, helps us as a group decide which of these should we move forward with first. So that's more or less sort of the scope of this. It's a pretty big enterprise, and it's not like typical science is done because we have to, again, be able to get the animals generated and again try to release them within 6 months, basically, of QC. Same thing with all the data generated. So it brings up some unique challenges for us in terms of just making sure we have everything streamlined.
Right.
Get that out.
Imagine the organization alone. So 4 different locations. How many people approximately?
I've never actually counted. It's a lot. I've been trying to think of my slide at the end. I have most of the people listed. That's probably at least 30 people, 40 people, somewhere in that neighborhood.
I imagine they're the supervisors of all of this stuff too. So you probably have people working.
Yeah, so we have lots of different meetings. And actually, this week at the Society for Neuroscience, we have an external advisory board, which is comprised of, and you would recognize all the names on our external advisory board, leading scientists and clinicians. And so we're meeting on Saturday this week for a full day of interaction and their assessments of sort of how we're doing.
Imagine you have to do a lot of Skype meetings in between.
Yes, we do a lot of meetings. Thankfully, the technology has gotten so much better. We're now on the Zoom technology, which is just absolutely fantastic. So we've had 12 different people on the phone, and it really works pretty well. And that you can share, anybody can share their screen. So it allows you to sort of fairly quickly sort of share data across sites. So that's actually worked reasonably well. And the other thing is, I think the people that we've come together are again really good to work with, and I think that's for these big consortia. That's the key, because again, it has to be people who are all committed to it, who all have the same level of investment, or otherwise, it just sort of falls apart. But thankfully, we have a great.
Yeah, it's an excellent team. So the center you're heading up in Indiana University, correct me if I'm wrong, is Model Organism Development and Evaluation for Late Onset AD. Right. The great acronym is Model-AD, for anyone who wants to look it up.
Yeah, and we have a website too. It's modelad.org.
Okay, I will definitely toss that in the show notes so anyone can later. I notice it says model organism. Now, whenever I think of AD models, or Alzheimer's disease models, I think of mice. One question: why mice? Two, are there any other organisms you're prepping these studies?
That's a great question. Yeah, I mean, that name was very definitely intentional.
Yep. How long did it take you to come up with that name?
It did take a little while. Our original name, I want to tell you, it was so horrible. Acronym that it would take you longer to say the acronym than the words, I think. So we've at the acronym. So the organisms. So we had to make some decisions. Again, we had a finite budget, and we decided we wanted to make the most number of models possible. That was really what was going to benefit rather than generating one model on multiple different organisms, for example. And so we decided, well, first that we would focus on the mouse, just because, again, I think largely that's where, again, I think we'll have the most impact. I mean, in terms of the researchers that are out there, there's so many that are using mice already to study basic biology and preclinical studies and all the rest. But even within the mice, we had to make some difficult decisions. For example, there's many different types of mouse strains, genetic background. We had to figure out, okay, how do we handle that? What's the right strain to use? And we decided just to move forward with C57 Black 6, and largely because that's what the community is using. Not necessarily, wasn't because that's the model we should be in. And I think
well-known mouse,
right? But we don't know for certain, and so we now actually have just recently got a supplement to start looking at genetic diversity, and looking at an impact of models we've already generated.
Right.
So that will come online. But the other was sort of the species. So we got a smaller supplement last year to start looking at some rat models. And so the model that we're first characterizing is an early onset model, just because we wanted to just run it through the pipeline and make sure everything works smoothly. But we have the plans, and the NIH seems potentially interested in expanding that into a far greater number of late onset rat models. And I think there's certainly certain advantages to the rats. One is they're much better for things like imaging, just because their brain is bigger, substantially bigger than the mouse. So I think you can get much better resolution of imaging data. I think the rat generally is better for some behavioral tasks and some electrophysiology, again, largely because they're bigger. But then we've had some discussions with other groups about, are there other species that we should be thinking about? There are some other animal models that have been out there for a while. We heard yesterday from Elizabeth Head, and she didn't talk about it, but she did a lot of work on the dog as a model of Alzheimer's disease, and they certainly developed some of the pathology, the agu and naked mole rat, and there's all kinds of different animals out there. But I think what we felt was that we would almost be best to sort of do a hierarchical sort of development of models, that we would have, first pass, it says, in the mouse, and tries to do as many as we can, as fast as we can, and identify the most promising, and maybe move those forward into the rat. And does that also work as well in the rat?
Right.
If it does, then move that forward into maybe even a non-human primate or another species, sort of to get us even other species, because I think that's, when I talk to the folks in pharma, that's really what they would like. They would like to have a series of models that get closer and closer to these before they get in.
Yeah, and I think mice are very appealing just because you can do things on much faster timescales, right? You can do many more experiments in one cohort of mice than you could ever in monkeys.
Yeah, but it also is the downside of then it's not the normal time frame of development of pathologies and things like that, and that's something we don't know what the right answer is,
whether or not it reflects the true disease.
Because if the disease takes, which it seems like it does, of course, 20-year preclinical window and things are changing in the brain, and then another 10-year clinical window, that's 30 years basically disease has been. So I think obviously we can't compress that into in a reasonable way, I would argue, probably to animals. Although, again, people before, it's interesting to look back at history, because, again, I've been in the field a long time. There were people who early on said, mice will never develop amyloid pathology. In fact, I remember getting grant reviews back, basically almost explicitly stating this, that they don't live long enough, they'll never develop that pathology.
Yeah.
And obviously, again, that's not true. It was, yes, figuring out what mutations. But I think that, again, we'll figure this out, I think, as time goes on.
So what got you into the field of Alzheimer's disease research?
Yeah, it's actually also interesting. Actually, it was similar to what Liz Head was interested in. I was in a lab as a postdoc at Johns Hopkins where we were interested in Down syndrome and the link to Alzheimer's disease.
Right.
And at the time, we just, the genetic location of APP had just been figured out. That it was on chromosome 12.
And that's the protein that creates amyloid beta fragments, right?
Right. Yeah. So the idea was, and what she clearly talked about yesterday, is that an extra copy of this gene generates amyloid was enough to then drive amyloid pathology and everything downstream of that. And so my first efforts were to generate mice that basically had sort of a genomic overexpression of APP. So we introduced the entire locus of that gene, huge, it's about almost 200 kilobase artificial chromosome, into the mouse gene, generate a mouse that basically overexpresses it. And that was sort of my first of all the different models that we've generated over the years. And so that's really where my interest came from. Now, it's also, as a lot of the students were describing yesterday, it's now much more of a personal interest, for me and my family, and my mother and my father-in-law both affected with dementia.
Sorry to hear that. Devastating.
But again, it's unfortunate. I mean, that's just the reality. I think of our people are living, which is great, living to be into their 80s and beyond. But the rates once you get of dementia, once that group is so high.
Yeah, and I think that's what makes it such a fascinating disease as well. Is we're all at risk, but we don't exactly know all of the factors that put us somewhere on the spectrum, right. Of risk, and so. These models will, I think, go such a long way in helping determine what are the causal elements, and then how do we fix those causal elements exactly. So, Yeah.
So that's very much our goal, is to help the field, and it's a very different era too, because I think what that does, I mean, when we're starting to see it, I think because it used to be an animal model field, that everybody developed their own models, and then yes, they might share them, but they might not.
Yeah.
And so it led to sort of a weird dynamic. And so I think it'll be interesting to see how this evolves. Is, if we're making all these animals available, then probably it's going to want everybody to make their animals available. At least if you want your animal to be utilized,
right? Exactly.
Then
why not?
Have to.
And it's all on a uniform platform. It's all on the same platform, the C57 black mouse, right?
Right now it is, yeah.
And so that's a big challenge for me when I'm reading these papers is, oh, this mouse is in one background, and this one mouse has black fur, this one mouse has white fur, they look completely different, this one mouse has brown fur, and I don't know how all of these other genetic factors
are and we have some data that suggests actually that genetic background does impact is probably likely to impact phenotypes.
Right.
We've done, for example, we've just done some initial screening where some of the same ApoE, ApoE4 mouse we've generated, we've looked at this in different genetic backgrounds now, and different strain combinations. It's clear that there's some strain combinations which shift, for example, the transcriptomics to sort of look more human-like. I mean, that some of the same networks and modules are activated than they are, for example, in C57 black 6. So that might be one that you would sort of say, well, maybe for ApoE we should be in that genetic background.
Well, thank you so much, Dr. Lamb, for coming to talk to us here. This is your first BrightFocus?
It is, yeah. So I've really enjoyed it. It's certainly nice to be here in a sunny, sunny spot for a few days, and and I think it's great. The other thing I wanted to say is that I appreciate what you're doing.
Thank you.
And I think it's really, really important to sort of have scientists be able to speak to the community. I think it's a really critical thing, and I don't think it's done nearly enough. So
yeah, I mean, well, everyone's busy, and I totally understand that. So it's great that I'm able to find the time here, because we were worried we wouldn't have the time or the space to do this kind of stuff. Anything else you'd love to say to the audience? Or?
No, I mean, just keep supporting research. I think that's what's going to finally get us there. And yes, we've had some failures, but I think that just makes, at least for me more determined that we can't afford not to do this. Really, at the end of the day.
Yeah. can't afford to give up at this point. So
Yeah.
thank you so much again,
Sure.
Dr. Lim.
All right, thanks.
That's going to be it here for us at the Straight From Scientist podcast. Thank you so much for listening, and especially if you provided feedback or are a sponsor of the podcast. If you'd like to become a supporter, you can go to patreon.com/SFS podcast.